All right, our next unit is animal physiology. Animal physiology. So the word physiology is a word that basically means we're studying both the structure, which is traditionally known as anatomy, and the function, which is, I guess, what you could call physiology. So we're looking at how things are made, the anatomy, all the parts, but also what they do. And the two tend to go together. All right. So first of all, we'll do a little bit of a review of some of the pertinent information that you should be familiar with at this point from grade 10 and grade 9 science. Mostly grade 10. Let's talk about cells. The cells are, of course, the primary building blocks of life, and the function of a cell mimics the function of organisms. Well, in fact, I should say it the other way around. A large organism like a human is actually sort of mimicking, or I guess you could say performing all the same jobs that cells have to perform in order to stay alive, but it's doing it in a cooperative way. And so you'll remember that cells come in all shapes and sizes, but each cell basically has to perform all the jobs of life. All the jobs of life. That's what I like to call all the things that have to happen in order to be alive. All the jobs of life. So, for instance, what are the jobs of life? Obtaining nutrients. And of course, the other key job of life, for most things anyway, is obtaining oxygen. Because these two things are basically what go together to produce the energy that allows life to exist. From a physics point of view, life is really simply just a bunch of energy changes that are happening one after the other or in conjunction with each other. And there are other functions. There's waste removal, detoxification. There are substances out there that can be toxic or poisonous, so we have to protect from those, and et cetera, et cetera. There's lots of functions. We're not going to be able to study them all. We're going to look primarily at how nutrients are obtained through the process of digestion, and we'll also look briefly at how oxygen is obtained through respiration, and maybe we'll see how much time we have and we can get to some of the others. All right, so back to cells. Well, cells do all these jobs, and in order to do that, they have to have a sort of a level of sophistication. So we often draw in grade 10 a typical animal cell, which, looks something like this. We draw a squiggly membrane. Okay, and we're going to have to put some labels on here, but it's going to get a little bit tight, so maybe I can color code things. So this would be the cell membrane, and we'll look at how that functions and what it's made of a little bit later on. Most animal cells, at least the ones that are making up organisms, have a nucleus somewhere, usually near the center. Let me do a different color on this one, just so that we can try to keep everything clear. So I'll use a red pen and draw the nucleus like this. And over here we can write nucleus. My carrot isn't going to make nucleus. N-U-C-L-E-U-S. And within the nucleus, there's often a sort of a dark spot, which I'll put also in red because it's very close to it. It's the nucleolus, the nucleolus. Okay, and we'll talk more about each of these things and their functions later. Another common feature of a cell is this membranous sort of structure, which we call the endoplasmic reticulum. Now, the problem with drawing cells is, of course, first of all, we're drawing a two-dimensional picture, and a cell is more 3D, so it's hard to express that. And secondly, cells are very full of stuff. When we draw the picture, they look like they're mostly empty with these little things floating around. But if we draw it all the way it's supposed to be, the picture becomes way too complicated. So this endoplasmic reticulum is everywhere in the cell, all over the place, but we only draw a few spots of it so that we can have room for other things. So endo come on, carrot. Endoplasmic reticulum. See, I just sharpened the carrot, and I've discovered that when you just sharpen it, it's almost too damp and fresh. So it doesn't work as well. Endoplasmic reticulum. Now, there's another structure that's found often associated with this endoplasmic reticulum, and they're very small structures. They're like little tiny dots, which I'll place sort of all around here, and they're usually found near the nucleus. And they're found within the endoplasmic reticulum, but only near the nucleus. And so sometimes we differentiate. We call this the rough endoplasmic reticulum, and the part that's away from the nucleus without these structures is called the smooth endoplasmic reticulum. And these structures can also exist outside, but they're found primarily in the endoplasmic reticulum near the nucleus, and they are called ribosomes. Ribosomes. Little blue dots. All right. Another important part of an animal cell. Let's see, what colors have I got? Well, let's go to black again. Another important part of an animal cell is these little tiny sausage-shaped organelles. And they have it's going to be hard to draw. They have a wiggly membrane inside of them. If I drew one up close, they look more like this, and then they have a membrane that wiggles like this all inside. But obviously that's too big for this picture, so we won't do it like that. We'll just draw it as best we can. And of course, these little sausage shapes are the mitochondria. One is called a mitochondrion. Many are called mitochondria. So let's see if I can write that in here. Since I'm only pointing to one, I'll call it mitochondrion. And again, this crazy carrot doesn't want to work today. So just give me a second. I'll see if I can't write that a little bit neater for you. It also doesn't work when you try to write too small. There we go. Mitochondrion. Right there. Okay. There are also some structures that we find in animal cells that are used for reproduction. I'll do them in yellow. They are little sort of weird shaped things like this. There's usually two of them, and they sort of stick together until it's time for the cell to reproduce, and then they split apart and they migrate to the ends of the cells in the process of meiosis or mitosis, and this is called the centrioles. The two of them together like that. That's an important part. Let's see. The other thing that cells have are all kinds of tiny little circles or vacuoles. Maybe I'll draw them in blue. They're little spaces. They're little membrane-bound bits and pieces, and usually they're fairly small, right? Some of them can be relatively large, but animal cells have lots of them, but they're very small. And these vacuoles are just basically spaces. And the cell has many different types of these vacuoles. So what happens, of course, is these are just pieces of cell membrane that have formed little pockets inside the cell. And inside these vacuoles, many different things can happen. Sometimes the cell has trapped food in there. particles of food that it's going to digest. And when it does that, the vacuole is called a lysosome for breaking down things. There are also vacuoles that break down toxins. For instance, there's something called a peroxisome, which breaks down the dangerous hydrogen peroxide if a cell were to encounter it. But they're all over the place, and they have several different jobs. But typically what they are is pieces of cell membrane that have sort of become pinched off and turned into little holes or little pockets all around the cell that do various things. Okay, and we'll talk more about all that. In fact, in grade 10, I'm sure you went through all this, and I'm sure you talked about what each of these things do. I'm sure you said that the nucleus is the brains of the cell, the mitochondrion was the powerhouse of the cell, and you will learn a little bit more about how they actually work if you take grade 11 and then grade 12 biology. In grade 12, we delve deeply into things like the mitochondrion in animals and the chloroplasts in plants, and we look at the complex chemistry of what's happening there. But for now, we just want to get an idea. Okay, so here's our cell, typical animal cell. Now the problem is that when we say typical, there's very few animal cells that actually look exactly like this. Typical just means this is sort of a general picture, a mishmash of all the different cells put together so we can label it. But the truth is that cells take on many different shapes and sizes. For instance, if we just go to Google here and we Google cells, we can see, let's do some images really quickly. You get the standard pictures that we just drew. But look at all these other pictures. They don't look anything like the one we drew. They all look strange. For instance, here in the top left, these are blood cells, red blood cells. They look like little donuts. And then we see all kinds of different shapes and sizes. In fact, if I specifically search, watch this, a neuron, which is a nerve cell. We get a picture of a nerve. Well, look at that. That doesn't look like anything that we just drew. And in fact, it doesn't, because it's got a very highly evolved function. And so what happens is typical cells that sort of live by themselves, they tend to look like the picture we drew. But most cells in a human body or any kind of organism have done something called specialization, where they sort of pick a specific job to be an expert at. And they give up all the other jobs, right? Cell specialization. And so we get all these weird looking cells. Here's a liver cell. And you'll see it looks kind of normalish. These are actual liver cells under microscopes. Also notice that under a microscope, a light microscope, we don't get to see very many of the organelles. They're all very small. Mostly we see the nucleus, and the rest just looks like a grainy haze. Here's an actual picture, but this looks to me like a drawing, not an actual picture of a cell. That's a cell that somebody has drawn, right? The actual pictures are these reddish ones that have been stained, and of course all these organelles are very small. How about a bone cell? Watch this, you'll see. Bones actually have cells. Very strange looking, not the same as the picture we've drawn. They're all very different. Let's do one more here. Let's do bone cell, liver cell, nerve cell, neuron. Let's do a muscle cell. Again, these are structures of muscles. The actual cells are not really... they're showing me, this are muscle fibers, which what the cells make up. But this picture here in the top right is a diagram of a muscle cell. It doesn't look anything like our picture, does it? So the reason for that is because most cells specialize. Cell specialization is a very important concept. And what it means cells adopt a particular function and often abandon other functions. Whoops, let's spell again. Let's abandon other functions. So I want you to think about kind of like the idea of a bunch of people lost on a desert island. Plane crashes and the survivors are gathered around. Now, Survival requires lots of jobs to get done, right? Their shelter has to be built, wood has to be found for fire, food has to be found, somebody has to collect berries, somebody has to work on the SOS signal, and so on. Somebody has to find water. And if all the people just run around trying to do everything, not much gets done very efficiently. So immediately what happens is the people who are trapped on the island begin to divide the jobs up. So one person is looking after the fire. They become specialized. They're not going to worry about food. Nothing about food, just fire. Another person will find food. Another person will find water. Another person will build a shelter, and so on. The person who builds the shelter doesn't waste their time looking for water. That's somebody else's job. So in an organism with many cells, the cells themselves can specialize into particular jobs. So some cells become specialized. So if we're talking about cells now and not people on an island, some cells become specialists at, maybe defense. Your immune system is full of cells that help defend against invaders. Some cells become specialized as messaging cells, like your neurons. Messengers. In fact, neurons are so highly specialized that they can't do any of the other jobs of life very well, and they would die on their own. That's how special they are. They've gotten so specific, they can't live alone, and every neuron has a special little cell that lives right beside it called a nurse cell that does all the other jobs for it. Because sometimes specialization comes at the expense of losing the ability to do other things. So messengers, defense, nutrient processing, right? Absorption of nutrients and all of that stuff. Producing different bodily fluids, secreting things, secretion of different bodily fluids that are necessary. Some cells do that really well. Liver cells do that really well. Some cells form structures like bone cells. They absorb calcium and they form complex structural elements. So you see, the cells are all picking different jobs. And this happens quite early on in the development of an embryo. At first, you're just a single cell, a fertilized cell, a sperm and an egg joined together, but then that cell begins to divide and divide and divide. And after, 50 or 60 divisions, the cells start to actually begin to choose specific jobs. Skin cells, for instance, have chosen to become long membranous, or form membranous sheets that basically protect us from the outside world and hold us all together, and so on. There's so many different jobs. And then, of course, once these cells have selected their different jobs, so groups of cells with the same job are called, they tend to group together, right? So they can all help each other out. They're called tissues. Tissues are groups of cells all performing the same job. So liver tissue are all cells found inside livers that are doing the same job. Perhaps they're detoxifying alcohol or whatever that might be. And then, of course, when you put several tissues together, tissues go on together to group together to do or to become organs. So you'll remember that from grade 10. Cells, tissues, and organs. That was the bulk of what you learned about in grade 10 biology. Organs are groups of tissues working together to perform something. Because some jobs are all on their own, right? But so the person building the shelter building is like a tissue. But many different tissues might get together to build the shelter. For instance, there could be wood and rock finders. There could be people mixing mud for mortar. There could be other people. So all of these different jobs are grouped together. So a tissue is like a single job. An organ may have several people with related jobs that all perform the same function. And then sometimes you'll get organs that work together in complex systems, organ systems, where everything is working together because now the job is so complex it needs many different things to happen. So you'll remember from grade 10 this sort of hierarchy of cells, which then specialize to become tissues, and tissues can become organs, and organs can become organ systems. Okay. Now, the other thing I want to talk about is some of the specific structures. We're not going to get into all the cell structures, but there's one structure on a cell that is probably the most important one, believe it or not. in terms of how a cell performs its job. And it's often the one we ignore. When we draw our cell, if I go back to my picture, I'll show you which one it was. It was the very first one, this guy right here. And it's just a stupid line around the outside. And we go, yeah, it just holds the cell together. Now, the cell membrane is the most critical part of a cell because it's what allows a cell to do almost everything that it does. It's so important. And so I want to take some time to talk specifically about the cell membrane because this often gets left out. And what I've discovered is that understanding how a cell membrane works answers so many questions that you may have as we go through this unit and on into the next years. So what does a cell membrane look like actually? We draw a single little line, but if we magnify that line, we find out it has a very complex structure. And I'm going to draw you a picture. The structure is made up, now of course, I don't want to get into too much detail because it'll make the video too long, but each of these structures that we're going to draw are really just bunches of atoms stuck together. And essentially, carbon atoms, hydrogen atoms, oxygen atoms, and nitrogen atoms, the odd sulfur atom here and there. That's primarily all the atoms that things are made of. Then there's little bits of other things, like there's little bits of iron inside of our bodies, Fe, there's magnesium, and there's a few calcium. But these are small amounts. These are by far the most. And out of these, the first three are by far the most common atoms you're made of. So what happens, of course, is we learned in grade 9 and 10 that these atoms will get together in different patterns and arrangements to fill their outer orbits and form molecules of all kinds. And so here are some of the molecules that they form. They form something called a phospholipid molecule, right? So I'll just, I guess I can erase that so it's out of our way. They form a phospholipid molecule, which is a complex bunch of C's, O's, and H's, and a little bit of P, a little bit of phosphorus, all stuck together. And they form a little shape that looks like a little circle. I'm going to get my pen back. A little circle with two long tails on it. And these little circles with two long tails stick together like this. Now, okay, remember, all these little lines represent are strings and groups of C's and O's and H's and atoms that are all stuck together. And then every so often, well, there's also another layer on the bottom. And the reason for this has to do with the fact that these molecules don't often like to be in the water. Part of them like the water and part of them don't. Then I'll take a red molecule, and I'm going to draw this as a big swirly sort of chunk like this, because that's what it is. This is a protein. And a protein is basically, if you were to look at one under a microscope, it looks very much like a big ball of string. That's kind of what it's made of. Okay? And it's sort of stuck in the middle of the membrane here. And then we have a little bit more of this little layer of molecules like this. Some of the proteins go all the way through, but some of them, like this one I'm going to draw next, are just sort of on one side. So here's another protein that's just kind of bubbled up here, and it's kind of sticking like here, outside like that. So only on one side of the membrane. Okay. We call this structure a phospholipid bilayer. Phospholipid, right? I can write that underneath here if you want. That's what these black things are. Phospholipid. And we'll learn more about their structure a little bit later on. And we call it a bilayer because you can see how it's made of two layers, one on top of the other. And then it's got these proteins. So these protein molecules that are stuck in between. That's what these red things are. Now, the proteins have very interesting jobs. What they do is they form passageways in and out of the membrane. So you see this one protein here that I have right through the middle. That allows things to squeeze through. And in fact, what a protein, imagine a ball of string. If you push it and twist it, it changes its shape. And it will be possible for you to stick your finger on a ball of string, wiggle your finger, and it would just kind of wiggle right through the ball of string. You could sort of make a hole in the middle. So things can go in and out of these. They're called protein gates. So they function as ways to let things in and out of a membrane. And these gates can be very complicated. They can be controlled by the cell to let some things in and some things out. And that's very important, right? When we talk about absorbing insulin or glucose or different molecules inside of us, what we mean is that those molecules are being passed through these special gates on the cell membrane. Now there are other molecules here. I'm going to erase some of these words to make room for the other pictures. All right, and I'll erase these arrows that I drew so that doesn't look too complex. There. Okay. Now, attached to now, the other thing is it's hard to draw, but you have to envision this as a 3D picture. So you have to kind of envision that it's not like a line. It's more like a giant sheet that stretches like this. If I draw a piece, okay, and then I'm going to draw these fast. What I'd like to do is I just tend to scribble. These are the little circles, just done faster. And then I go like this in between, right? Here are the little lines in between. I'm just doing a quicker job. And maybe there's a big protein sort of right there, and it wiggles over here, and it sticks out on this side, stuck in there on that side. There might be another protein sort of stuck over here that we can see sticking out, right? And maybe one more on this side sticking out of here. And all across the top are these little balls. All across the top like this. So you see the membrane is 3D. Just drawing it fast. And so everywhere you have these proteins, you can have things going in and out. Now we're not done. I'm also going to erase our title because I need to draw up there a little bit. I'll go in blue here. There are little chains of carbon and hydrogen atoms that stick out, and oh shoot, I want to erase. Okay, back to blue. They stick out like this, and they look like little branched trees. Some of them are attached right onto the black sort of the black molecules, the phospholipids, and some of them are attached to the protein molecules like this. And they're all sticky all over the place. And some are on the outside, and some are on the inside. And these are called, because of they're made of hydrogen and carbon mostly. They're called, when they stick to a protein, they're called a glycoprotein. And when they stick to a lipid, a black molecule, they're called a glycolipid. Whoops. Glycolipid. But they stick all over. And you know what? They are very interesting too, because what they form are like little receiver places. Other molecules can float by. Here's a molecule. So I'll give you an example. A molecule of insulin. Here's a molecule of insulin, right? Now, what happens is you've just eaten a big dinner. So what you have are all these little molecules of sugar, glucose, and they're floating around. And they have to get inside the cell. They have to get through the membrane. Glucose is a fairly large molecule, so it's stuck out here. So your body makes this yellow guy called insulin. Insulin comes over here, and he lands on the receptor site. He fits just perfectly. It's all about the shape, right? Molecules in biology are all about if they have the right shape and they can stick together. And when he does that, he sort of twists or distorts this little tree into a slightly different shape, which distorts the little red protein molecule, which causes a hole to open up, which allows the glucose now to shoot through there and come out on the other side and go into the cell. And that's a complex process. And I'm really fast forwarding here, just giving you a brief idea so when we talk about digestion and absorbing nutrients in the intestines, this is what's happening. This is what's going on. The nutrients are being sucked through the cell membranes, through these protein gates. And sometimes they'll just slide right through. Other times they need a molecule like insulin to turn the gate on or open it. It's very complicated, but it's kind of a neat process. And you need to understand that so that you can understand what we mean when we say that food is absorbed through the small intestine, right? Okay, and this model of our cell membrane that we've drawn, it has a name. It's called the fluid mosaic. It's a mosaic because we recognize that the membrane is made of many different structures, all blended together. The fluid mosaic model. And of course it's called fluid because it's actually liquidy. These little molecules, they're not really stuck together very strongly, so they can wiggle and move. In fact, this protein right here can shift and wiggle its way through these molecules and then end up somewhere over here. It's fluid. That's what that means. The molecules are not trapped in one spot. They can interact. So it's actually like its own little living thing almost, the membrane. So you can see how drawing a simple line and calling it cell membrane is really not doing this special structure very much justice. So I wanted to highlight that because that's kind of a critical part of how cells actually function. Because the membrane is how the cell interacts with its outside world, right? And the outside world, say this area up here, and the inside of the cell, down below, down here, are very different places. And the cell needs to be very selective in how it lets things go in and out in order for life processes to work. And what you're going to find if you go on in biology and you study biochemistry, a lot of biochemistry happens on a membrane of some kind, whether it's a cell membrane or the membrane inside a mitochondria or the membrane inside a chloroplast. Membranes are actually the most important cell structures in my mind because every bit of biochemistry pretty much relies on membranes to happen. So there you go. One little bit of detail that you might have not covered in grade 10. So we reviewed cells. We see cell specialization. We see cells turning into tissues, tissues turning into organs and organ systems. And of course, we have now seen a little bit about the cell membrane itself and how complex that is. So we'll stop there. In our next video, we'll look at some of these molecules that are moving around in and out of the cell and how they are critically